Hello everyone, my name is Cassandra Springer and today I will be talking about the Nova Labs. So just a quick overview of the program itself. It is a free digital platform for students and other learners that uses games and interactives to explore scientific ideas. There are multiple games for different types of learning. It's mostly scientific stuff and computer learning more so than English and things like that. But anywhere from solar storms to evolution, cybersecurity, RNA, energy systems, and more. The Nova Labs allow learners to conduct their own research on topics through visual aids, analysis, and by being able to use the same data that experts use. Each Nova Lab looks into different areas of present and relative research. Each lab has informational videos so that learners can follow along and answer scientific questions or help come up with answers to today's problems. The Nova Labs have 3 different groups that make this program possible. There are the platform partners. Some of them include Design Squad Nation, Digital Learning Day, Khan Academy, PBS Learning Media, Science Buddies, and more. The platform advisors is made up a team of 13, and the lab team is 6 directors and 14 student advisors. And so not only are there adults and experts on the team to create this program, there are also students who are also involved to help, which I think is super cool because they kind of know what students are looking for in programs so that they can help it be appealing to students while also working on creating a program to possibly help researchers find answers to unsolved problems today. The Nova Labs are also funded by Argosy Foundation, Biogen, Lockheed Martin, and NASA. So the lab that I picked is the RNA Virtual Lab. This is what the page will look like once you pull it up. And a little bit more about this lab itself is it's a extension of a game called Eterna, which I will get into later towards the end of this video. But by playing this game, you can actually help scientists understand an important family of biomolecules called RNA. And it has you go through different levels that gradually get more and more complicated until you create a whole working system. So was not able to find a video of someone else showing how to use this site. So this is just the introductory video to the RNA game.
Where does life come from? This is one of the most important questions humanity has ever posed, and the scientific answer is, we don't entirely know. You might think that cracking DNA's genetic code should have explained life's origins, and it definitely helped. Thanks to our understanding of DNA, we can map out the history of evolution all the way back to single-celled life. But that's where we're stuck. The problem is, DNA is a great way to store information, but it doesn't do much else. Cells rely on other molecules like proteins to replicate, grow, and survive. Proteins, on the other hand, work great as molecular machines to keep cells alive and healthy, but they can't store information or copy themselves. They need DNA for that. So we have a chicken and egg problem. DNA needs proteins to function, and proteins need DNA to exist. So which came first? Which molecule made life possible? Well, there's a third type of molecule that may hold the answer: RNA. Most scientists think that RNA came first because RNA can do 2 jobs: store information and perform various functions that keep cells alive. This idea that RNA came first is called the RNA world hypothesis. RNA world suggests that billions of years ago, in some primordial soup of molecules, a self-replicating RNA formed. This may have happened in volcanic vents deep on the ocean floor, or perhaps clay clumps brought the necessary chemical building blocks together. Some scientists have even speculated that early RNAs formed on Mars and hitched a ride on an asteroid to our planet. One way or another, self-replicating RNAs emerged, multiplied, and evolved. Over millions of years, they developed into a legion of molecular machines. These microscopic proto-life forms blossomed and competed. The best collections of code lived on, and the weaker ones died out. Survival of the fittest was the name of the game. This competition for survival eventually led RNAs to evolve the ability to build strong, stable proteins, which excelled at carrying out complex biological processes. And somewhere along the line, some critical RNAs mutated into the familiar double helix of DNA. DNA became a stable archive of genetic information that stored blueprints for the most successful RNA and protein molecules. Life became more complex over trillions of tiny steps and happy accidents. And all the while, the RNA lineup grew alongside lengthening genomes of DNA and complex proteins. And it's all still happening inside your body. RNAs have adapted to become the Swiss Army knives of our cells. Today, they can slice, dice, catalyze, build, destroy, code, replicate, and transform. A remarkable diversity from the simplest of beginnings, a single self-replicating RNA molecule.
So by playing, and I know this video is only supposed to be under 2 minutes, but I just thought it was a good educational video to kind of introduce some of the students to what RNA is and how it kind of works within our body systems. And by playing this version of the game, you'll be able to discover that molecular RNA machines carry out a wide variety of essential functions in our body, and you will get to kind of play around and build those. So when you go to the main page, you will be able to play the game, and you can sign up with Google or a guest pass, but if you do guest, it will not be saved. So just a quick sign in with Google. And then you can go ahead and play game. And it's just going to have you start off with the basics, so it's kind of going to walk you through what trials 1, 2, and 3 are going to be about. So then you can click next. And then there's always going to be a little direction box on your screen to kind of guide you on what your mission is going to be about. So if you click next, it'll tell you what you kind of need to do. There's 4 bases, and it walks you through what each of the bases are. And down here is how we can change the colors to each of the bases. So my first mission is to have 4 or more G's or red dots on the RNA. So I click the red in the palette and I just fill in each of the spaces. And then I have cleared the round. And then I would go on to do the next mission. So you continue to do all of those missions, and then I went ahead and finished all of them. So once you get to the last one and once you finish this last round, you have finished the game, and then you can go on to join the Eterna game, where that is where you can dive deeper into research with RNA, which again I will talk about in a minute. So this is the lab handout that I would give with all of the instructions. So you would go to pbs.org for the RNA lab and click play game. You can, like I said before, you can create an account or sign in as a guest, but if you do the guest, your progress will not be saved. And clicking the video icon will pop up the introduction video that I showed you earlier in this presentation. Once you have finished, you can start the tutorial mode, which will walk you through how to play the game just like I had showed you. So starting puzzle 1 until you have cleared the game. Once you have completed all 6 puzzles, you have completed the tutorial mode and can move on to trial 1. And it gets more complex as you go. Start trial 1 and complete the round. Once you have finished, you can add your TNRA to your virtual cell. Continue working through trial 1 until you have completed all 6 levels and have added your RNA pieces to the cell, and then you can move on to trial 2. After you completed the 6 trials in trial 2, you can move on to 3. And once you've completed the 6 trials in 3, you're finished, and you can continue and join Eterna, which allows you to deeper explore today's problems. And in Eterna, you can actually send in your RNA folds and what you've created for a competition, and the winning RNAs actually get looked at by experts to see if it would fold the way that you have predicted it would fold. And so that's just pretty cool that your work can be looked at by other people as well. So a couple classroom applications. You can use the video lessons and quizzes by going through video lessons with your students or having them work in groups to do it. Once they have completed, there are quizzes available that you can have your students take just to get a sense of their understanding and see what maybe you have to talk more about in class or work with. Homework, you could assign the puzzles to be worked on at home or in class. In-class discussions, after watching the videos, you can facilitate a classroom discussion about what the students learned or found interesting about the video. And you could also incorporate concept maps by having the students make a concept map of topics addressed in the videos or things they learned, or have them create a concept map of how to create an RNA. So I really liked this application. It's something that I could see myself using in my classroom to allow the students to move at their own pace, working through these, and if they wanted to continue on their research in RNA development.